Hi, welcome back. Today we are going to continue on a slightly different topic, but one that I keep touching upon. I have mentioned diffusion again and again. Today we're going to dive straight into the question of diffusion. I'm going to talk to you a little bit in terms of a brief background, and then we'll dive into details of the physical theory of diffusion, how it relates to thermodynamics, and how that connects to cell biology and quantitative biology, which then becomes what we keep calling cellular biophysics. So, we are kind of familiar with diffusion, but you will be surprised to learn that it is critical for the thing that we do right now, breathe. So, some of you maybe practice yoga and some of you watch yoga on TV, it's quite popular these days. This process of inhalation, exhalation is driving air, even while I talk, into our lungs. The lungs are nothing but branched networks of cells of tissue with air gaps, and at the smallest cavity of the air gaps there is diffusive exchange of gases in our lungs. So, in that sense, understanding diffusion is not just something obscure and basic science and we do not care, but something fundamental to life itself, our life, human life, and we value human life, and we value our good health, and coronavirus is an aerially transmitted disease. So, we wear masks normally because we want to protect our airways, because this virus attacks our airways, right. So, another very common process in which diffusion is critical at a cellular scale is our neuronal system, and ion diffusion, which both occurs through channels that are dedicated for the transport of ions, those are ion channels, as well as passive transport through the membrane itself, diffusion through the membrane. Both are responsible for controlling our neuronal activity. So, these are crucial factors, and therefore, understanding the physical basis of it, because it is a physical process, becomes critical, right. So, in some senses, passive transport is related to the process by which without channels also ions can move in. Opening and closing of ion channels can of course regulate their flow, and concentration gradients are the key driver in diffusion of ions from inside to out or outside to in. But they are also responsible for the spread of ions, because even when there is no concentration gradient, ions are still going to be moving around and spreading uniformly. In a sense, you could argue that diffusion forms the basis of transport at a cellular level, and we are all familiar with those beautiful movies of molecular motors walking, and we will have a chance to talk about them in the later segments of this course. But diffusion is an important and almost often ignored aspect in cell biology of how things move around. So, for example, an RNA polymerase that is present in one part of the cell can move to the other parts within about 1 to 10 seconds purely by diffusion, purely by the same random walk that we think particles of air that are around us that are performing. DNA on with a polymerase that is transcribing it translocates in similar times, but that is not passive transport. We know that this is active transport. So, we argue that active and passive transport, diffusive transport, have similar time scales, right. Longer time scales like seconds, tens of seconds, minutes, we see directed motion, and one of these classic examples is E. coli motion. And if you remember in the Reynolds number segment, we talked a lot about low Reynolds numbers, the rule of drag, the nature of random walk of E. coli. The funny part is that E. coli is small enough, 2 microns in length, 1 micron in diameter, that in fact, it is subject to thermal motion and will undergo diffusive motion in short time scales, but in long time scales it undergoes vectorial motion. And so, what a little bit of history of random walks is important here to highlight that Robert Brown made a series of microscopic observations, and I think this is an important lesson, because sometimes your curiosity to just know what is there is such an important driver in scientific discovery, and you don't need to know too much history. So sometimes I would suggest, if you have a microscope, please go to a pond or a little stream, or if you live near the ocean, then the river, or in the well that you take your water from, and look under the microscope. Just look, because you will be fascinated. I am fascinated every time I have the chance to do this. And Robert Brown in 1828 was fascinated. He wrote his observations in terms of a brief account of microscopic observations. So this is the second part of scientific discovery, by the way. Write down what you see. Try to explain. Maybe it is just for yourself. Maybe, no teacher is going to look at it. No professor has time to read your diary, but write it, because you never know when you will be the new discoverer, the new Brown. Thomas Graham, also the inventor of dialysis, discovered the spontaneous diffusion of gases, the one that I was talking about, which is involved in the exchange of gases in your lung. Adolf Fick, who came up with Fick's laws of diffusion, 1855, medical physics, mixing of air in lungs, heart and mechanics, is another pioneer in the theory of random walks. And Darcy in 1856 worked on the flow of water through sand bed. So these are some of the people who have been major contributors, and unfortunately you see it's all 1800s. So in some senses these are old theories, but old theories that work, right? This is not about history or geography or which country or something. It's just simply, if you do the same experiments, you'll get the same answers, and that is the beauty of science, right? But strangely, you'll be surprised to know that random walk and diffusion theory is also used in finance. So if you want to make a lot of money, stock markets are your option. Harshad Mehta, number 2. These transitions of the stock market prices, I think I've taken them from Yahoo Finance some years ago. This was 9 a.m. in I think 2021. Show these fluctuations which almost resemble stochastic fluctuations that come out of random walks, and random walk models have been very successful in explaining the kind of behavior that we see in these statistical data. Indeed, George Gao said something very beautiful about random walks. He said Brownian motion is unique amongst physical processes because it makes visible the constant state of internal restlessness of bodies in the absence of any external cause. It is a weakened and remote testimony of thermal molecular motions. This is such a eloquent sentence, such a well written sentence, because in fact it illustrates the fact that if you, as I said, take a microscope and look, it doesn't matter whether there are animals or insects or no living things in it, you will see motion. And this motion is not because you were thumping the table or your dog was coming around and barking at you. It is just simply because of heat. The ambient 30 degrees, 40 degrees, depending on the temperature in your town or city or your lab, that is the cause of the motion. And this is really because it's elegant, because this is pure energy, right? It's around us everywhere, right? Now, so when I said random walks in finance, Louis Bachelier in 1900s published his PhD thesis in mathematics. It is called The Theory of Speculation. I don't know the French words for it. This was indeed the first attempt at using the laws of random walks and diffusion to mathematical prediction of stock market values. We don't know how successful he was financially, but certainly he set the stage for a lot of what are called quants or quantitative mathematical approaches in mathematical finances. And we come to 1905 to 1908. So you remember my earlier description of luminaries of contributors to diffusion and random walks were from the 1800s. Now we are 1905, 1908, and we come to Albert Einstein. Albert Einstein was, as you know, a Jewish researcher in Berlin. He was made to leave, or he was frightened of his life, so he left because there was political persecution in Germany. He went to Switzerland, where he worked as a patent attorney. And in that strange time, he and his wife, who was herself a mathematician, they worked out a series of papers. This is 1905 to 1908 period. The first paper is translated as about the molecular kinetic theory of heat and its consequent motion in fluids that are at rest of suspended particles. In other words, on the movement of suspended particles in a stationary liquid demanded by the kinetic theory of kinetic molecular theory of heat. This is published in a journal that you will never have heard of. It's not Nature, Science, Cell or Phys Rev Letters. It's called Annalen der Physik. It doesn't exist anymore as far as I know in that old form, because it was a German journal. He then went on to write a paper on the theory of Brownian movement, which is also Annalen der Physik, and eine neue Bestimmung der Molekulardimensionen, a new determination of molecular dimensions, also in the same journal, 1906. So you see, this is a very productive time when he was working as a patent attorney and patent examiner in the Swiss patent office in Bern in Switzerland, which means that you can do research, excellent research, groundbreaking research, even if you don't have all the means to it. Of course, theory is easy because you don't need a lab. When you need a lab, things are a bit trickier, but remember that you have to look beyond your own limitations, and we live in a country, India, we have high contrast. Sometimes we have very fancy labs, sometimes we have no labs. Rather than complain about it, try to find a way around it, okay? And hopefully you will get better. The final paper in this series was Elementary Theory of Brownian Motion, which was published in 1908 in the Zeitschrift für Elektrochemie, Journal for Electrochemistry. My only point of bringing this up is the fact that these theories are more than 110 years, 115 years old, and they're still relevant, and they make sense to us, and they help us explain a lot of things. And secondly, that provenance of the journals where the work is published is not as important as the provenance of the work itself, and it is what we refer to as the test of time, right? So if the work is valid, then it will remain, whether it is Archimedes, whether it is Sushruth, whether it is Albert Einstein, right? So we are interested in the the microscopic theory that underlies diffusion. When we observe particles, when Robert Brown observed particles moving around, he knew that they were moving, but he didn't know why. In fact, the first idea was that they are alive. So he went and took coal particles. He took a candle and made a soot out of it, took those soot particles, suspended them in water, looked at them under the microscope, and he saw they were moving. So soot is not alive, we know that, but he said, oh, maybe some insect got in the way. So he went to a friend who went to Egypt, to the pyramids, and he took a brick from the pyramids and crushed it. Of course, that is 4,000, 5,000 years old. That must not be alive now. There must be nothing living in it. And he looked under the microscope and it still moved. So he said, oh, this is crazy. Something keeps moving even if it's dead for thousands of years. How is that possible? And that how is that possible is in a way answered by Einstein's work, because individual particles require to be thought of as individual particles. Random motion is due to thermal energy, and this thermal energy is given by T, which is the absolute temperature in Kelvin, and Kb, which is Boltzmann's constant, and mass of the particle and velocity. So in order to get an answer to this question, we need to infer the RMS, root mean square velocity, the average velocity that a particle undergoes due to this thermal motion. In one dimension, the velocity along x we can call Vx, and the kinetic energy Ke. So Ke from thermodynamics we know in every direction x, y and z is equal to half mv in the case of x square. Yeah. So if you have that and you know that the kinetic energy is half kbt in one dimension, then we can show that Vx square is equal to kbt, or in other words, the average root mean square velocity of a particle in one dimension is under root of kbt by m, kbt being the thermal energy and m being the mass of the object, which means that we can now calculate this. For example, lysozyme, which has a gram molecular weight of 1.4 into 10 to the power 4 grams, that is 14,000 grams, and the number of moles, number of molecules in a mole is 6 into 10 to the power 23, that is Avogadro's number. So the molecular mass is 10 to the power minus 20 grams with prefactor 2.3, and at 300 Kelvin, when we substitute the values of 4.14 into 10 to the power minus 14 grams centimeter square per second square of kbt, we get a value which is something I would like you to calculate as a part of your exercises. So for the next steps, we are going to talk about random walks in higher dimensions, shape of particle spread distribution, number of steps, Gaussian distribution, Perrin's experiment. Thank you very much.